Hi, my name is Kang Igon, and today I'm going to talk about um, my subject, the exosome containing lipK3 inducing the cancer cell death by necroptosis. Cancer is a diverse collection of life-threatening disease caused by abnormal and invasive cell proliferation. There are about 40 million new cases of cancer in adults each year, and half of these people will die from the disease. Tumor is used to describe tissue where cells are multiplying abnormally. Benign tumors are encapsulated and localized in size, but in contrast, malignant tumors can continually increase their size by breaking through basal lamina and invading adjacent tissues. Cancer is the disease caused by malignant tumor, as you know. After antigens are processed by immature dendritic cells, in short IMDC, they are presented to T cells by mature dendritic cells through the major histocompatibility. Complex class 1 or MHC class 2 complexes binding to CD8 or CD4 T cells separately. Simultaneously, the mature dendritic cells also express cytokines such as interferon gamma and interleukin 12 to synergistically stimulate the T cells. CD8 T cells further differentiate into cytotoxic T lymphocytes, and CD4 T cells further differentiate into interferon gamma secreting T helper 1 cells to assist in activating the CD8 cells and other innate immune cells such as natural killer cells, granulocytes or macrophages to directly kill the tumor cells. Um, to solve the problem of cancer, I used the exosome mechanism. Extracellular microvasculars, MV, and exosomes are the major types of extracellular vesicles, in short EV, which can carry and transport molecular messages, such as proteins, messenger RNA, and microRNA. Thereby, EVs could serve to mark the changes of the producing cells and to function on the recipient cells. Mm, the traditionally, apoptosis is the only form of programmed cell death, and necroptosis is the passive death of cells under extreme conditions, independent of the regulation of intracellular and extracellular signals. Apoptosis and necroptosis have different morphological features. The former exhibits cell shrinkage, chromatin condensation, formation of apoptotic bodies, and phagocytosis by adjacent cells, and the latter exhibits increased cell volume, organelle shrinkage, plasma membrane disintegration, and other characteristics. More importantly, after necroptosis occurs, the cellular contents are released and cause an immune response. Then the damage-related pattern molecules, in short DAMP, such as HMGB1 and the mitochondrial DNA can be detected in the blood, which are markers of necroptosis. Uh, the necroptosis is regulated cell death which recurs various proteins such as tumor necrosis factor, in short TNF, or the lipK1, lipK3, and mixed lineage kinase domain like MLKL. Single LipK3 molecule needs to be oligomerized to activate MLKL and LipK1. These activations induce MLKLs to be oligomerized and translocated to the plasma membrane, which followed by necroptosis and cell death. Despite of that, lots of research show the mechanisms of necroptosis. It is hard to apply it in cancer therapy because many key regulators related with the necroptosis are generally downregulated in cancer cells. Exosomes can solve this problem. Exosomes are extracellular vesicles which participate in intracellular communication. And PD-1 expressed exosomes can target cancer cells very well. Exosome producer cells have proteins consist with PD-1, PTGFRN, MAPL, LipK3, and constitutive oligomerizing domain. Between the MAPL and LipK3, there is just linker which can be cleaved by 405 nanometer UV. Cleaved LipK3 form LipK3 trimer in exosomes and activate MLKL in targeted cancer cells. This study shows PD-1 expressed exosomes containing oligomerized LipK3 can target cancer cells and stimulate necroptosis. In this research, I will show the information of it by using 5 chapters. First, background, the explanation of species of programmed cell death and the reason of choosing the necroptosis as cancer therapy. And second, the overview of necroptosis, the molecular mechanism. Here we can show why we choose LipK3 protein as a key regulator. And third, I show the relevance of necroptosis in cancer. And fourth, detailed structure of proteins expressed on surface membrane of exosome producer cells and the way to synthesis. And fifth, I have to show the design of exosomes. Explanation of the process designing structure of exosome referring to the mechanisms of necroptosis. Exosomes must express PD-1 on their surface and contain the oligomerized LipK3. And the way I will use the way to this kind of data. First, to show the characterization of exosome, I'll show whether exosomes contain oligomerized LipK3 and BCA protein assay is proper way to show the data, I think. Also, how many MLKL formed oligomerized form in cancer cells need to be shown. And then I will show the necroptosis degree of cancer cells. There are many groups of cancer cells and or of other groups are treated with different materials as there will be a group of non-treated and bare exosome treated and LipK1 expressed exosomes and LipK3 expressed exosomes and MLKL expressed exosome and LipK1 and LipK3 expressed exosome and LipK3 and MLKL expressed exosomes and LipK1, LipK3 and MLKL expressed exosome. And the flow cytometry analysis is proper to check how many cancer cells died by the necroptosis. Thank you.